Dear students, please note down. These notes are available in the description of video. Please download your notes. Dear students, in this lecture of developmental biology, today we will clear gastrulation in chick. Dear students, as we know, in case of chick, in case of hen, the birds, the development begins inside the female reproductive tract in oviduct fallopian tube. The process of fertilization, that is internal, and after fertilization the zygote, the single celled embryo that converts into a multicellular embryo, the morula stage, and then morula converts into blastula. In case of chick, the blastula is discoblastula. At the time of egg laying, the embryo is blastula or discoblastula. This process, gastrulation, it begins after egg laying. When incubation starts, just after 4-5 hours of incubation, this process, gastrulation, it begins. And this gastrulation in chick involves following steps. Step number first, that is formation of endoderm or hypoblast. Dear student, the first step, it is essential. The formation of endoderm or hypoblast, that involves in the digestion of yolk. It means endoderm or hypoblast provides nourishment to the developing embryo by digesting the yolk material. And after the formation of endoderm or hypoblast, the second step is formation of primitive streak, that is caudal end of embryo, that represents the tail region of embryo. This second step, which is formation of primitive streak. Dear students, these are different prospective areas which we have discussed in fate map of chick embryo. This is epidermal ectoderm, extra embryonic ectoderm. It is neuroectoderm. This area is notochord. Below this, precaudal mesoderm. Then these are somatic mesoderm. This area is lateral plate mesoderm. And dear students, this diagram shows early stage of formation of primitive streak. The formation of primitive streak occurs due to the specific movements of mesodermal cells and notochord cell. It means after the formation of endoderm, the prospective mesoderm and prospective notochord cells start to migrate towards the mid-dorsal line. As a result, this longitudinal thickening appears, and this longitudinal thickening appears in the center line, midline of body, that is called primitive streak. And this primitive streak represents caudal end of embryo. It is tail region of embryo. Note down, this primitive streak encloses a groove in its central region, and that groove is primitive groove. And in its anterior region, this primitive streak possesses an elevation, and this node-like structure, it is named primitive knot, or it is Hansen's knot. Note down, competitive exam students, this primitive streak which develops due to specific movements of prospective areas of mesoderm and notochord cells. As a result, this longitudinal thickening which appears in middle part of embryo, this thickening represents primitive streak, and it encloses a groove, that is primitive groove, and in its anterior region, a small elevation, a node is present, that is primitive knot, or it is Hansen's knot. This primitive streak, it is homologous to the lateral lips of blastopore of amphibian. while this Hansen's knot, this primitive knot, it is homologous to the dorsal lip of blastopore. Dear students, in the formation of this primitive streak, the specific movements of prospective embryonic and extra-embryonic areas, and these movements are of three types. Note down, in the formation of primitive streak, the specific movements of these mesodermal cells and notochord cells, and these movements are of three types. Competitive exam students, these movements are convergence, invagination and divergence. These are three types of movements: convergence, invagination and divergence. First type of movements, convergence. These movements are inward movements of prospective areas towards the mid dorsal line of body. Convergence. The embryo, after the formation of hypoblast, it is two layered. This outer layer, that is epiblast, and this one is hypoblast, or it is endoderm. In convergence, the prospective areas of mesoderm, and these cells migrate, they show movements inwardly towards this mid dorsal line. These movements are convergence. Second type of movements, these are invagination. Dear students, invagination means inward sinking of prospective areas. From this mid dorsal line, these cells show inward movement. These are sinking movements of the prospective areas. Third type of movements are divergence. And in divergence, the movements of prospective areas, the cells outwardly. And these outward movements of prospective areas called divergence. Dear students, in case of gastrulation, object, first step is formation of endoderm, that is hypoblast. This outer layer, that is epiblast. From this epiblast, the lowermost layer of cells delaminate. As a result, this layer is formed. It is hypoblast, it is endoderm. And the formation of this hypoblast, which is essential, because this hypoblast that involves in the digestion of yolk, it means this hypoblast provides nourishment to the developing embryo. And after the formation of this hypoblast endoderm, the second step is formation of primitive streak. And this primitive streak involves the movements of mesodermal cells and notochord cells towards the mid dorsal line. And these movements are of three types: convergence, sinking movements, these are invagination, and these outward movements are divergence. As a result, in this center line, mid dorsal line, a longitudinal thickening appears, and that is primitive streak, which represents the caudal region of embryo, the tail region of embryo, and it appears just after 7-8 hours of incubation. In this anterior region, this primitive streak possesses a small elevation, and this knot is, it is primitive knot, or it is Hansen's knot. Dear students, this primitive streak, it is homologous to lateral lips of blastopore of amphibians, while this Hansen knot, that is homologous to dorsal lip of blastopore.